Every MCAT passage that mentions cells is really testing one thing: can you predict how molecules move? If you've ever mixed up diffusion, facilitated diffusion, and active transport under pressure, this is where it clicks. In this video, we'll break down the fluid mosaic model, membrane permeability, and the transport mechanisms that show up again and again on exam day. By the end, you'll be able to look at a passage and instantly know what's crossing the membrane, how, and why. No guessing, just strategy. The fluid mosaic model describes the cell membrane as a dynamic phospholipid bilayer with embedded proteins, cholesterol, and carbohydrates that regulate structure and function. Think of the membrane as a sea of lipids with proteins floating like boats, constantly moving laterally. This fluidity is crucial for processes like signaling and transport. Each phospholipid is amphipathic, meaning it has a hydrophilic head and hydrophobic tails, which drives spontaneous bilayer formation in aqueous environments, an example of entropy-driven self-assembly that also connects to thermodynamics on the MCAT. Cholesterol intercalates between phospholipids and acts as a fluidity buffer. At high temperatures, it stabilizes the membrane, and at low temperatures, it prevents tight packing, maintaining flexibility. Proteins serve diverse roles, including transporters, receptors, enzymes, and anchors to the cytoskeleton, while carbohydrates attached to lipids or proteins form glycolipids and glycoproteins involved in cell recognition, a concept often tested alongside immune system topics. A useful mnemonic is LPC. Lipids form the barrier, proteins do the work, carbohydrates identify the cell. On the MCAT, you may be asked how changes in temperature or lipid composition affect membrane fluidity, or how disrupting cholesterol impacts permeability and protein function. Understanding that the membrane is not rigid but a fluid mosaic helps you reason through these passage-based questions. Selective permeability of the cell membrane allows small non-polar molecules to diffuse freely while restricting ions and large polar molecules. The hydrophobic core of the phospholipid bilayer is the key barrier. Non-polar molecules like oxygen and carbon dioxide dissolve easily in this lipid environment and diffuse across without assistance. In contrast, charged species such as sodium or chloride ions and large polar molecules like glucose cannot pass directly because they would have to shed their hydration shells and interact unfavorably with the hydrophobic interior. A helpful way to remember this is like dissolves like. Nonpolar crosses nonpolar. Water is a special case. Although polar, it can diffuse slowly and is greatly facilitated by aquaporin channels. On the MCAT, this concept often appears in questions about permeability rankings or predicting which molecules require transport proteins. For example, if given a list of molecules O2 glucose, Na+, you should immediately recognize that O2 diffuses freely, glucose needs facilitated diffusion, and Na+ typically requires channels or pumps. This idea also ties into pharmacology. Lipid-soluble drugs cross membranes more easily, influencing absorption and distribution. Understanding selective permeability helps you interpret experimental data on membrane transport rates and reason through how cells maintain distinct internal environments. Passive transport includes simple diffusion, facilitated diffusion, and osmosis, all of which move substances down their concentration gradients without energy input. Simple diffusion involves molecules moving directly through the lipid bilayer from high to low concentration, driven by kinetic energy and entropy. Facilitated diffusion, on the other hand, requires membrane proteins, such as channels or carriers, to help polar or charged substances cross, but it still does not require ATP. Channels provide a pore, often gated by voltage or ligands, while carriers undergo conformational changes to shuttle molecules across. Osmosis is the movement of water across a semi-permeable membrane, typically through aquaporins, from low solute concentration to high solute concentration. A useful mnemonic is water follows solute. On the MCAT, you will often see questions about tonicity, hypotonic, isotonic, and hypertonic solutions, and their effects on cells, such as swelling or shrinking. Another common test angle is distinguishing between simple and facilitated diffusion based on saturation kinetics. Facilitated diffusion can become saturated because it depends on a finite number of transport proteins. Connecting this to enzyme kinetics can help. Both show a maximum rate when all proteins are occupied. Recognizing these patterns allows you to quickly interpret graphs and experimental data involving transport rates. Active transport uses energy, often from ATP, to move substances against their concentration gradients via pumps and coupled transporters. The classic example is the sodium-potassium pump, which moves 3 sodium ions out of the cell and 2 potassium ions into the cell per ATP, hydrolyzed, creating both concentration gradients and an electrochemical gradient essential for nerve impulses. A helpful mnemonic is 3 out, 2 inches ATP wins. Primary active transport directly uses ATP, while secondary active transport uses the energy stored in ion gradients established by primary transporters. In secondary transport, you will encounter symporters, which move 2 substances in the same direction, and antiporters, which move them in opposite directions. On the MCAT, this often appears in passages about glucose uptake in the intestines via a sodium-glucose symporter or proton gradients in mitochondria linked to oxidative phosphorylation. Understanding active transport also connects to membrane potential, electrochemistry, and cellular energetics. Questions may ask you to predict what happens if ATP is depleted or if a pump is inhibited. Typically, gradients collapse and downstream processes fail. Being comfortable with these mechanisms allows you to reason through both discrete and passage-based questions involving cellular homeostasis. Vesicular transport mechanisms such as endocytosis and exocytosis move large molecules and particles across the membrane using membrane remodeling. Endocytosis brings material into the cell and includes phagocytosis for large particles, pinocytosis for fluids, and receptor-mediated endocytosis for specific molecules like LDL cholesterol. In receptor-mediated endocytosis, ligands bind to receptors, triggering vesicle formation, a process often associated with clathrin-coated pits. Exocytosis is the reverse process, where vesicles fuse with the membrane to release contents outside the cell, such as neurotransmitters or hormones. A useful way to remember this is endo enters, exo exits. These processes require energy and involve the cytoskeleton and membrane curvature proteins. On the MCAT, you may be asked to identify which transport mechanism is being used based on the size of the cargo or the presence of receptors. Vesicular transport also connects to topics like protein trafficking in the endomembrane system, including the ER and Golgi apparatus, and to cell signaling in neurotransmission. Understanding how cells move bulk materials helps you integrate knowledge across multiple systems and tackle complex passage-based questions with confidence.